Hi, DrBiology here, and this video is about protein synthesis. So what I'm going to cover is transcription and translation for the AQA A-level biology spec. I'll be looking at the role of RNA, mRNA, and also talking about how proteins are made. I hope you enjoy the video. Okay, so let's look at protein synthesis. So here we have a cell. Inside the nucleus is the DNA, and also on the outside you can see a ribosome. Okay, so first of all, let's talk about the production of mRNA in transcription. So during transcription, an mRNA copy of a gene is made from DNA. In eukaryotic cells, transcription takes place in the nucleus. Transcription starts when RNA polymerase attaches to the DNA double helix at the beginning of a gene. The hydrogen bonds between the two DNA strands in the gene break, separating the strands, and the DNA molecule uncoils at that point, exposing some of the bases. One of the strands is then used as a template to make an mRNA copy. Free RNA nucleotides alongside the exposed bases are attracted to these bases, and specific complementary base pairing occurs, meaning the mRNA strand ends up being a complementary copy of the DNA template strand, except the base T is replaced by U in RNA. So eventually an mRNA strand is being produced. The hydrogen bonds between the uncoiled strands of the DNA, they then reform once the RNA polymerase has passed by and the strands coil back into a double helix. So now we have our piece of RNA, but at the moment it is known as pre-mRNA because in eukaryotes they contain both exons that code for the production of proteins and introns that don't. So these have to be spliced out and removed. So the introns are removed in a process called splicing before the code can pass out of the nucleus. mRNA then moves out of the nucleus through the nuclear pore and attaches to a ribosome in the cytoplasm. So the mRNA, it attaches to a ribosome and a start codon then is read, which is usually AUG, which is the first codon. So before I talk about translation, we need to talk about tRNA. It basically carries the amino acids that are used to make proteins to the ribosomes. So tRNA is a single polynucleotide strand that's folded into a clover shape. Hydrogen bonds between specific base pairs hold the molecule in this shape. Every tRNA molecule has a specific sequence of 3 bases at one end, and these are called anticodons. They also have an amino acid binding site at the other end. So firstly, mRNA attaches to the ribosome, start codon must be read, and then second, the next tRNA molecule moves in and matches with the mRNA codon, so the anticodon and codon are complementary, while the amino acids form a peptide bond. So the previous tRNA detaches and the mRNA shifts and moves the next tRNA molecule to come in. The protein grows, so you get more and more amino acids until you get a polypeptide. So now the protein is formed and it's ready to finish folding to become a 3D tertiary structure to become a functional protein. So how does the triplet code for amino acids? Well, let's have a look at this table. So here we have an mRNA code and we need to look at each triplet code. So let's look at the first letter A, and then second letter is U, third letter is G, and that gives us the start codon, which is codes for methionine. The next one, so the first letter is C, second letter is C, third letter is C, so that means that the amino acid is proline. And the next one, GGC, and that will give us glycine. And then UAA, so UAA, and that gives us the stop codon. I hope you found this video useful. Please do subscribe to DrBiology. I'll see you soon.